Hello students, we have started with chapter 3 Inheritance and Variation. In the first part we have studied genetics, then the history of scientist Gregor John Mendel, who is called as father of genetics. Then why Mendel selected pea plants for his experiment, we see those reasons. Then we studied the steps involved in the Mendel's breeding experiments. Now in this part 2 we will study some genetic terminologies. First genetic terminology is character. Character is a specific feature of an organism. For example, height of stem. Students, in the last part we have studied the characters shown by pea plant. 7 contrasting characters shown by pea plant. In that we have studied height of stem, then the color of flower, position of flower on the pea plant, shape of the seed, color of the seed, and so on. These are called as characters. The second terminology is trait. Trait, it is nothing but an inherited character and its detectable variant, that is whether the plant is tall or dwarf, whether the plant produces yellow or green seeds is called as trait. Next is factor. Factor, it is the unit of heredity. Factors are the particles which are present in an organism and which are responsible for inheritance and expression of a character. Factors can pass from one generation to the next generation through gametes. Factors determines a biological or genetical character of an organism. Next is gene. Gene is a segment of DNA which is responsible for inheritance and expression of that character. We have studied this definition, that is of alleles or allelomorphs. Alleles or allelomorphs are the two or more alternative forms of a given gene or a factor. Alleles occupy identical or same positions on homologous chromosomes. Dominant, this is a form of a allele. Dominant allele that expresses its trait even in the presence of an alternative allele, that is in homozygous heterozygous condition only. Alternately, you can define dominant trait as, or the allele as, the allele that expresses itself in F1 generation is called as dominant, or the allele which masks the expression of other allele in F1 generation. Next is recessive. It is allele which is not expressed in the presence of an alternative allele in heterozygous condition. It is only expresses in homozygous condition. Or recessive, it is an allele that does not express in F1 hybrid. Phenotype, it is the external appearance of an individual for any trait. Example, in a pea plant, for the height of stem, tall and the dwarf are the two phenotypes. Tall is determined by capital T or capital T small t, and dwarfness is represented by small t. Genotype. Genotype, it is genetic constitution or genetic makeup of an organism with respect to particular trait. It is the representation of genetic constitution of an individual with respect to single character or a set of characters. For example, pea tall plants can have a genotype either capital T or capital T small t, and dwarf plants have genotype small t small t. Homozygous. An individual possesses identical alleles. Same alleles are present. Those individuals are called as homozygous or pure for a particular trait. Homozygous plants always breeds true to the trait and they produces only one kinds of gametes. For example, tall pea plants produces only capital T and dwarf plants produces only small t gametes. Heterozygous. These are heterozygous and individual possessing contrasting alleles for a particular trait. Heterozygous does not breed true for a trait and they produces two types of gametes. For example, F1 generation hybrids C capital T and small t is an heterozygous individual which is also called as hybrid. Pure lines, these are the individuals or a groups of individuals population which is homozygous or true breeding for one or more traits. The plants which breeds true for a particular trait is called as pure line plant. It is a descendant of a single homozygous parent produced after self-fertilization. Next is monohybrid. Monohybrid, this is again a hybrid or the heterozygous individual which is heterozygous for a single trait and is produced from a cross between two pure parents differing in single pair of contrasting character. For example, capital T small t is a hybrid tall plant. It is produced from a cross between pure tall, that is capital T and pure dwarf small t. F1 generation is also called as first filial generation. It consists of all the offsprings produced from a parental cross alternatively. F2 generation is a second filial generation or progeny produced from selfing of F1 generation. For example, it is the progeny which is produced from two F1 individuals, capital T small t crossed with capital T small t. Punnett square or is also called as checkers board. Punnett is the name of scientist who discovered this diagrammatic representation. So, it is a diagrammatic representation of a particular cross. It helps to predict the progeny of a cross. This table also represents different permutations and combinations of fertilization between gametes of opposite mating types. Homologous chromosomes. Students, we have studied this terminology with the help of diagram in part 1. So, homologous chromosomes, these are morphologically, genetically and structurally identical chromosomes which are present in a diploid cell. Homologous chromosomes undergoes synapse or crossing over during meiosis that leads to recombination. Backcross. It is a cross of F1 progeny with any one of the parents. For example, when F1 plant is crossed back with pure tall plant or pure dwarf plant. It can be shown like this. Capital T small t is crossed back with capital T that is pure tall, or small t that is pure dwarf. Test cross. It is a cross of F1 progeny with homozygous recessive parent. That is, the F1 plant crossed back with the pure dwarf plant small t. It's called as test cross. Students, test cross can be a back cross, but back cross is not necessarily a test cross. Test cross, it is used in plant breeding experiments to test the homozygous or heterozygous nature of a hybrid. Phenotypic ratio. So, it is the ratio of offsprings produced in F2 and subsequent generation with respect to their physical appearance. For example, phenotypic ratio in the monohybrid cross comes to 3:1. 3 tall is to 1 dwarf. Genotypic ratio. It is the ratio of offsprings produced in F2 and subsequent generation with respect to their genetic makeup. For example, it can be represented as 1 capital T that is pure tall is to 2 capital T small t. Hybrid tall is to 1 small t. Pure dwarf is the F2 genotypic ratio in monohybrid cross. So phenotypic ratio is 3:1, tall is to dwarf, and genotypic ratio is capital T is to 2, capital T is to small t. Monohybrid cross. It is a cross between two pure parents differing in one pair of contrasting character or only one heritable trait. It's called as monohybrid cross. Example, it can be a cross between a pure tall and pure dwarf plants. Dihybrid cross. It is a cross between parents differing in two heritable traits. It's called as dihybrid cross. So two characters are considered at a time in dihybrid cross. For example, it is a cross between pure tall and round seeded plant with pure dwarf and wrinkle seeded plant. Okay students, so for today this only. In the next part we'll study in detail monohybrid cross, dihybrid cross and test cross experiments with suitable example.